Okay, great. Thanks, Nikolai. I'm very happy to be here to talk to you today, and I just want to try the idea is just to give you a flavor of what we have been doing with the EvoSep instrument in the last year and a half or so. So I'm now moved to Belfast, but only just since 1 month, so I have nothing there, no lab, no nothing. So all the data I'm going to be showing is essentially from Zurich. And so, yeah, this is the menu. So I will try to make one or two just sort of brief comments about what the motivation is to go this direction. I think this is probably kind of understood, but and then the first data set I'd like to show is kind of the first evaluation that we made of the EvoSep platform. This is a mouse model that we're interested in, and there we were able to do this measurement both on a kind of more standard nano LC platform and also the EvoSep. So it's kind of easy to make a comparison and sort of see what the advantages might be of the EvoSep type method. Something we're also very interested is the kind of global analysis of protein complex using kind of protein correlation profiling type methods. Rudy talked a bit about this on Sunday, I guess, and the question is whether can short gradients help with this. And we've also been involved in a collaboration to work on this diaPASEF method, and I'll mention a little bit at the end some kind of early data on this, but this is sort of relatively preliminary, I'd say, and still quite some work to do there. So what about the motivation? So actually I realize now that Rudy talked quite a bit about this already if you were in the Brooker session actually on Monday, but I'll just kind of re-summarize it in kind of one slide for people who aren't there. This is actually work from Olga Vitek, who's a statistician at Northeastern University and a student in her group, Ting Huang. And essentially what they did was a bunch of simulations, and the question here was what if you have a sort of limited set of resources to do a certain kind of number of measurements and in a particular study, and they were talking about kind of clinical proteomics and biomarker studies for the most part here, would you rather measure a very large number of samples with a shorter gradient, or let's say measuring a smaller number of proteins, or would you like to measure using a more in-depth method but with a smaller number of samples? So what actually gets you more information out in the end? And they have done lots of simulations on this. So what they first do is they take a bunch of real world data of all kinds, all different types, and they essentially model the variance with respect to the protein abundance in the real data, right? So this is, we all know that the higher abundance proteins are easier to quantify more precisely, and the lower abundance proteins are a little bit more difficult, and so there's kind of a curve that says what that relationship is. So they model this relationship, and then they simulate a bunch of training and validation data sets that include sort of in silico biomarkers, right? So they inject some biomarkers into the data at various levels, either low abundance or high abundance, or kind of all across the abundance range. And then what they do is vary the number of quantified proteins. So they start with sort of 60, which might be the kind of SRM style experiment, or 500, 1,000, 3,000, and 5,000. And this is in sort of order of abundance, right? So these would be like the 60 most abundant proteins all the way down to let's say 5,000 proteins. And this is a typo. This should say the number of quantified subjects here. So then they also vary the number of biological samples that are in the study, anywhere from 10 all the way up to 400. And then they essentially use a random forest to try and make predictions about what are the correct experimental groups, but also what are the most predictive proteins for predicting those groups. Using a random forest. And the results essentially look something like this. So you have this matrix which shows this number here is the sensitivity of identifying so how many of these predictive proteins that they kind of injected in silico into the data set, how many of those can be recovered when they do the statistical analysis or the random forest analysis. And essentially the sort of the story here is that having more subjects is as you would expect always good. It's good to have more subjects for statistical power, but it's somewhat potentially counterintuitive and surprising is that as you increase the number of proteins, it's actually possible that the sensitivity goes down, and this is particularly true if you have a small number of subjects. So I think the kind of lesson here is that we need to increase this dimension of the data to have a lot of subjects, and that's especially true if we are increasing the number of proteins that we can detect and quantify, we need to at the same time increase the number of subjects, and if we have to make a choice between these things, then depending on the experimental design and so on, it might be worth thinking about it rather increasing the number of subjects as opposed to the number of proteins that detected. And I think this fits very well into the kind of EvoSep philosophy, which says what we should be trying to do is measure lots and lots of samples to get statistical power, even if this it may come at the loss of some depth of coverage. Another thing that I think is kind of intuitive is essentially as you decrease the gradient length, if you're doing DDA analysis, I think the prediction would be that it's likely that this is going to hurt you, because in DDA mode you need time to sample precursors and do MSMS spectra, and if you shrink the gradient then you're going to run out of time, and the expectation is that you're going to have precursor undersampling, and our guess is that this should be less prevalent in DIA mode, where you have a deterministic duty cycle, and essentially it shouldn't really matter. There still may be some losses due to shrinking the gradient due to kind of loss of selectivity or suppression effects, but it shouldn't have anything to do with precursor undersampling, and we've seen this in previous data before. If we take some standard lysate sample and we inject it in various modes using a 90-minute gradient or reducing to 60, 45 or 30 minutes, and we look at it in DDA mode or in DIA mode, either with 32 or 64 window precursor isolation windows, what you can see is that there's a fairly steep slope with the DDA analysis, but the slopes of the loss of sort of information with decreasing gradient when you essentially are using DA mode is much less steep. So there's still some losses. This is now protein level. There's more losses at the peptide level, but it seems like we can do a pretty good job in DA mode when we're shrinking the gradient of maintaining the information. I think this also came out very clearly in the first EvoSep paper, that it didn't escape them that it was a good idea to run DA mode with these short gradients. So that's the kind of the motivational part. Why did we actually go in this direction? So I'll now move on to this first kind of test data set that we generated in Zurich about a year ago now, I would say. And this is the model for this is this BXD family of mice, and this is currently the largest population, the largest and best characterized mouse genetic reference population. So essentially what they're doing is they're taking two laboratory strains of mice, and they cross them and produce a whole bunch of daughter strains, essentially, which are extremely well genetically characterized. So there's 183 different strains, all with different genomes that are a mixture of the two parental strains, and they know how these genes are segregated in terms of the parental genes. And this has been a kind of a test bed in our lab, and it comes mostly from the group of Johan Auwerks, but it's been a test bed in our lab for, eventually if you wanted to do real kind of clinical proteomics, it's going to be much more complex than this, but this is a kind of a simplified system that might represent what you may want to do in clinical proteomics. And the point of having this genetic reference population is that you can also do genetic association, either with the phenotypes of these mouse or more frequently now with the molecular quantities. So for example, like protein QTL analysis. So here's a study, and I should say that this is mostly work of Evan Williams in the group, and also Yibo Wu before him. So here's what the study looks like. They took 50 of these strains from this well-defined reference population strain. They feed them two different diets. So one is a sort of chow diet, one is a high-fat diet, and a large number of these die or live a natural lifespan. They also sacrifice them at certain points along the way. They collect many different tissues. There's a big focus on liver in this study because it's kind of metabolic. Also the study is related to metabolic disease, and they throw a whole bunch of technologies at them, genomics, transcriptomics, proteomics and metabolomics, and our involvement is the proteomics, of course. And so we had already acquired the data for about 400 of these mice using the SCI-X system in SWATH mode, using the exigent, kind of a standard nano LC with a 60-minute gradient. And we decided we would acquire the same set of data using the EvoSep method. So for these 400 injections that we needed to do, this took about 8 weeks of instrument time. And actually you'll notice that this 8 weeks is not 400 times the the gradient length, right, it takes a lot longer to actually measure these because of downtime and tuning and all the things that are essentially listed here. We had to change the LC column several times. We had to clean the machine. We had injector sample problems and so on. And this is the daily business of operating a nano LC. I think that most of nano LC-MS system that most of you will be familiar with. With the EvoSep system, we could acquire the same data set in roughly 9 days. We had some kind of minor crashes and some minor problems, but I think it was considerably easier to acquire the data with this system. We didn't have to clean the mass spectrometer in the middle of this run, whereas we did a couple of times up here, and we didn't have to change the LC column anywhere in the middle of the acquisition of these 400 or so injections. And so let's look at a few characteristics of the data. So the first one is the retention time stability. So we're always spiking in these retention time peptides, these IRT peptides. And you can see here the retention time stability for 2 of these peptides on the EvoSep as compared to this nano LC gradient on the exigent. And you can see that this is actually really nice and stable. Here it's a little bit more bouncy, and I think these kind of discrete shifts here correspond probably to change of the LC column. So this is quite nice. We can also look at the quantitative stability. So we include a bunch of technical replicates in the middle of the experiment. And so these are 2 adjacent technical replicates, right? So this is run 79 versus run 80, and this has very nice correlation. You can also say the same thing for a distant technical replicate sample. So this is run 79 versus run 406, and the quantitative the correlation seems to be quite good. This is now the correlation for different biological samples, which is a bit lower, but that's what you expect because they're different biological samples. And I can say that this data is basically very comparable to what we had with the longer gradient data. In terms of the number of peptides and proteins that we could quantify, so on this exigent 60-minute gradient, we're talking about 3,000 or so proteins from around 32,000 peptides. On the EvoSep, it was a bit less. Well, it was significantly less at the peptide level, but at the protein level, we maintain about sort of 90% of the coverage. And of course, this is a little bit older SciEx system, so the numbers are a bit lower here than you potentially could get with some of the newer equipment. But I think for the purpose of this comparison, doesn't really matter. And so essentially this is the number of detected peptides from the EvoSep and the exigent, which is relatively stable across time. We have some ones that drop out here, but these are all assigned to actually problems related to the sample processing for the most part. And so we were reasonably happy with this, that we maintain a large kind of most of the information at the protein level. And the last piece of data I want to show from this study is just to show that we can recover some kind of known biology. What I'm showing here is some peptides from this protein, this DHTK-KD1, which is known to be under the control of a particular allele that's different between these 2 different parental mice strains. And so if we look, the coloring shows you for each of these different strains whether the allele is from the one parent mouse strain or the other one, and so these are colored by red and blue. And essentially, what you can see is that this is a quantitative trait locus. So any mice that have this red allele have a high expression of this protein. Any mice that have the blue allele have a low expression of this protein. And essentially, the picture looks the same whether you look at either of the data sets. And this is the kind of thing that they're looking for in terms of really evaluating the quality of the data, because they have a lot of known true positive stuff that they know about this mice that they sort of expect to see. And we have a lot of examples where the genetic associations that we already know, where these alleles are controlling the expression of a particular protein, we could essentially recover these as well in the EvoSep data as we could in the exigent data, nano LC data. So the conclusion here from this kind of mouse part is that we have a really dramatic increase in throughput, and not just the kind of theoretical instrument time time, like the number of sample injections multiplied by the runtime. It's actually also everything in between that, including the downtime and the maintenance. It's, in our hands at least, is much improved. We have some kind of modest loss of information, at least at the protein level. We don't lose very much. I didn't actually show data on this, but it does seem like there are lesser batch effects, and so that means we don't need to apply such an aggressive batch correction or normalization, and this is obviously a benefit. And since then, we've actually conducted several more large-scale measurements of these in these mice studies. This is going on, so we've just collected like 400 samples from intestine and also from brain, I think. And the guy who's in charge of this project says there's no way he's kind of going back to the old way of doing things, because he's a biologist and he doesn't care about mass spectrometers and he just wants to get things done, essentially. So he's like he broke his heart to do the first study, and I think it's really kind of changing the way that we think about, to some extent, what we can get done. Right, this first time we did this study, that's a lot of planning to get sort of 8 weeks of measurement time and so on, and we're now in the position that if we want to measure like 400 samples, it can be done without much planning, and we can just sort of essentially do the measurement. I'm very interested in protein interactions, and something we like to do is this global analysis of protein complexes using these kind of protein correlation profiling methods. So this is also something I think Rudy mentioned a bit on Sunday maybe. The basic idea here is that we take some cells, we do a sort of a gentle lysis and that keeps the protein complexes intact. We fractionate these using size exclusion chromatography, or there are many different ways to fractionate the protein complexes, but essentially what you do is collect a large number of fractions, and this is usually something like 60 or 80 fractions, and then each of these fractions is subjected to standard kind of bottom-up mass spectrometry. And then what we do is we're quantifying the proteins across this size exclusion fractionation, and we reconstruct the protein profiles, and if there's sort of a coelution between these proteins, then we take that as evidence that these proteins were together in a protein complex at some point. And so essentially this is the standard sort of protein correlation profiling protocol, and we've sort of modified this a bit, or our version of it at least is using size exclusion chromatography, somewhat after what Leonard Foster has been working on in the last few years. We do 60 to 80 fractions. We use DIA mode to acquire the data, so that we can have kind of complete data sets where we are able to reconstruct these profiles across the size exclusion gradient. And essentially we've developed a new data analysis workflow which we call complex-centric analysis, that we think is increasing the sensitivity and selectivity in the protein complex detection. And so the question here is, can we actually increase the throughput? So the problem with this workflow is that it's currently ridiculously slow. If you have, for one biological sample, you have to acquire data from 70 fractions, and you're using sort of medium or long gradients, then you're talking about sort of something on the order of 10 days of measurement time for a single biological sample. And this makes it very, very difficult to do comparative studies where you want to do multiple conditions and you want to do replicates. And so the question is now, can we make this go a little bit faster using the EvoSep system? And so we decided to try this using the 60 samples per day method. And if we do that, then we can ideally acquire the data from one biological sample in about a day, or a little bit more than a day, which is, I don't know, you can't exactly call that high throughput either, but it's higher throughput at least. So the data set I'm going to show is also one that Rudy mentioned. This is this Hela cell comparison study, where they took Hela cells of different variants of Hela from all around the world, and they kind of checked whether they were the same or different. Basically, I mean, the summary was that they all looked quite different from each other. And we took basically 2 of these, and we the CCL variant, and we ran that in 3 biological replicates, and we took one of the Kyoto variants, and we also ran that in 3 biological replicates using this sort of fast SEC-SWATH analysis, where we can do kind of one day per biological sample. And the summary here is this. So this is the number of protein complexes, proteins with an SEC feature or protein SEC features. So this is slightly different because we sometimes see the same protein in multiple different SEC features, or the number of peptides we can detect. And the first column is the numbers from our original very first study, which came out about, I guess, a year ago or now. This is one replicate of HEC293 cells where we used 120-minute gradients, and we can see something on the order of 600 complexes. And what I'm comparing in the second column now is the data from this Hela variant comparison, where we compare the CCL2 and Kyoto variants, and there we're using this 60 per day, 21-minute gradient method from EvoSep, which allowed us to do 6 samples, essentially 2 biological conditions, these 2 variants, and in 3 replicates. And what you can see is that we are essentially covering more or less the same number of proteins, peptides, and so on. And in fact, what we actually really care about in this experiment is whether we can see the same number of protein complexes. And so you could say, well, this is a little bit unfair to compare 1 replicate here to 6 replicates here, but actually I think it's not unfair, because the amount of acquisition time that we need to use for this is actually more than we need to use for 6 replicates in this method. So I think it's quite a reasonable comparison in this case. And so I actually have a talk in the interactome session where I'm going to go a little bit more into the details of this, what the kind of biology results for, but I think for the purpose of this seminar, I think this is kind of the important result here. Okay. Last thing I want to talk about very quickly is this diaPASEF method. So this is a collaboration that we have ongoing also with Matthias Mann's lab. Florian Meyer is the lead guy here. Also Hannes Rust, who's the originator of the OpenSWATH software, and he's now adapting this to work with diaPASEF data, and we in Zurich and also Brooker were helping out this. So I just point out that there's a preprint which was put up a few months ago, that if you're interested you can take a look at that, and we're also working hard to try and get the actual paper out relatively soon. So I think that PASEF, and maybe even to some extent diaPASEF, has been described already at the meeting a few times, so I'm not going to go into too much detail about the trap time mobility separations or so on, but I just point out a few things that I think are important from the acquisition method. So the question is, like, what can we actually improve by combining PASEF and DIA? And I think there's a few important things here. One is this orthogonal separation of ion mobility, right? So we get an additional dimension of separation, so hopefully the sort of interferences should go down and the selectivity should go up. I think this was already in the regular PASEF mode. Also this ion focusing effect, so we get a little bit more sensitivity by compressing these ions into small packages in the trapped ion mobility system and releasing them into the mass spectrometer. We get a boost in terms of signal, and these two things, I think these were the advances that came in this PASEF paper from the Mann lab. But what the question is, what can we get beyond that by combining this with DIA? And I think what we can get is essentially an increase in the global efficiency of ion utilization in MS2 scans, and I'll say what that means in a little bit further. Essentially, in a standard DIA experiment, the efficiency or the duty cycle is 1 divided by the number of precursor windows, right? So in this sort of classical SWATH method, it would be something on the order of 3%, or even 1.5% if you're using the 64 window method. And just to point out that this is actually much worse for DDA, right? So DIA is already a significant kind of improvement in terms of ion utilization over more classical DDA methods. But the question is, can we increase this efficiency number by synchronizing the quadrupole with ion mobility separation? And this could potentially lead to increases in duty cycle speed. And also all the other benefits that come along with DIA, the kind of more deterministic quantification and so on. So this is what essentially the data looks like if you plot the mass versus the ion mobility dimension, and this is ignoring now the retention time dimension. In fact, all the retention time dimension is collapsed from a single run, is collapsed onto this. So this is the whole picture for one LCMS run. And what you see is that we get separation in the peptides, we get the +1 charge guys up here, and the sort of multiply charged peptide species are somewhere in this cloud. These are the 2+, the 3+ are usually a little bit further down. And essentially what the diaPASEF method does is to And so one thing to say is this is in this trapped ion mobility, we're starting at the top, right? So the ions come out of the trapped ion mobility device. The biggest ones come out first, and then the smaller ones kind of come out later on. And so what we do in this diaPASEF methods is at the start of this ion mobility cycle, we position the quadrupole in a kind of high mass range where we know that the ions should be coming out of the ion mobility device should be large at this point in time. And as time goes on in the ion mobility scan, we can shift the quadrupole over to smaller molecular weights. And the idea being here, as I mentioned, to really try to capture as much of the ion current as possible. And so the way I think you can think about this is essentially the ion mobility, you can think about this as time, right? So you have a sort of 100 millisecond cycle of the ion mobility device. The big stuff starts to come out first. You position the quadrupole here, and these blue lines are kind of representing MS2 scans that you're sampling. And now later on in the ion mobility cycle, we expect the ions to be over here, so we shift the quadrupole and we do more MS2 scans. And so essentially from the top to the bottom, we're acquiring about 1,000 top pushes during the course of this 100 millisecond ion mobility cycle, and essentially this is what this looks like from a method perspective. We also, as I mentioned, Hannes Ross did a lot of adaptations to the OpenSWATH software in order to really utilize this kind of additional dimension. And the way this works is that we now know what, in a library-based way, we know what the ion mobility of the peptides is likely to be. So that means in the DIA data, we can selectively extract the signal for those fragments from the ion mobility scans where we expect them to be. Right? So if this is again basically like this map here is the same one as here, and we essentially know that a given peptide should be in a certain space, so we can ignore this whole set of scans over here and this whole set of scans over here, and we selectively extract the signal where we expect it to be in the ion mobility space. And so just to show a few little bits of data, so most of the development was done on kind of longer gradient methods, but just recently we've started to try to combine this methodology with the EvoSep. I should point out as well that you would have the possibility to run actually very fast duty cycles with this kind of acquisition mode. So it's potentially a very interesting fit with the EvoSep, but we haven't really explored that in a lot of depth yet. And so essentially this is what the acquisition method looks like for this particular experiment. So we also have the possibility, I mean, in real life, the acquisition method, we're using smaller windows. This is kind of a schematic, but we're using smaller windows, and we don't always necessarily cover all of the precursors in one cycle of the ion mobility. So sometimes we're using multiple runs of the ion mobility to cover essentially all the precursors. So what the real world method looks like is something more like this. So this green bar represents the first cycle of the ion mobility device, and we have the quadrupole set here. And so as the ion mobility cycle goes, we have the quadrupole set in this position, and when we get down to here, we shift it over here and we capture these ions here. And then the next cycle, we get the next one. So we're essentially doing 2 precursor isolation windows per cycle of the ion mobility device. This is a little bit technical details. You don't need to worry too much about that. I think what's more important for the purpose of this is to say that this is now in a Hela, 150 nanograms of Hela lysate using the 60 samples per day method with the 21-minute gradient. We're detecting something on the order of 4,500 proteins with a kind of nice completeness in the data matrix. And this blue line is the accumulation line. So we're seeing essentially more or less the same set of proteins across all these samples. And I should say again that these proteins are inferred with proteotypic peptides, so we're not doing a sort of maximum parsimony sort of protein grouping step. So I think if you were to add protein grouping on top of this would potentially push the number up a little bit higher, but for the moment we prefer to use this kind of proteotypic peptide inferred data. And just to have a quick look at the quantitative aspects of this, we have just the standard things. Replicate 1 versus replicate 4 seems to have quite a nice correlation at the protein level. The CV is something on the range of what we would expect it to be, and this is the distribution of CVs for the whole thing. And so just I'll finish up pretty quickly. We've now, for the first time, tried to apply this diaPASEF method to what I discussed earlier on, this SEC size exclusion protein correlation profiling method. So we took one of these Hela cell lines, we fractionated it using size exclusion chromatography, and this is the sets of peptides and proteins that we can identify across the size exclusion chromatography. So we identify something on the order of 42,000 peptides and 5,700 proteins across the size exclusion gradient. Maybe I'll skip over this and just jump straight to the results, which is essentially that with this Hela CCL2 sample, by where we're using this SEC-SWATH or SEC-diaPASEF method, if you wish, we essentially have kind of similar, quite a similar set of results. We have a little bit more protein complexes using the diaPASEF method, which we're happy about, but actually also important is that we're using a lot less material for the diaPASEF method, which is going to be potentially useful in the future if we want to go to samples where we want to scale down. So I think I went a little bit longer than I wanted to there, but I'll try to summarize. I mean, essentially, we can go a lot faster, measure a lot more sample, and the important thing is that we get a lot more statistical power from this, and we're trying to overcome noise from this high biological variance studies. I think there's really good reasons to expect that gradient shortening should be less affected in DIA, and also now I think essentially quite some evidence for that also. We can analyze hundreds of samples per week, and the information loss that we get compared with the longer gradients is fairly modest, and this is shown in this kind of mouse study. For this kind of global protein complex analysis, we really badly need an increase in speed, and this seems to work pretty well. The preliminary diaPASEF data suggests that it's working out, and it should be a very good combination with shorter gradients, and this could actually be very important for this SEC-SWATH method or this SEC method, where we would like to get the runtime down from 1 day to much less than 1 day. So we'd like to, something we definitely want to investigate is the ability of kind of diaPASEF to be combined with these even shorter EvoSep gradients. But again, there's a lot of work to be done there. So the people who did the work were primarily in this mouse study, Evan Williams and Claudia Martelli in Zurich. This kind of complex-centric workflow, PCP workflow, was spearheaded by Moritz Heusel and later Isabel Bluda, who worked on the data analysis side, who has subsequently gone on to Matthias Mann's lab, actually. And this fast complex-centric method was work primarily from Claudia Martelli, Peng Zhu, Isabel Bluda, and George Rosenberger. And this is the people I mentioned already who are involved in this diaPASEF workflow. So I'm going to leave it there. I'm sorry if I went a little bit possibly over time, and yeah, thanks for your attention. I'll answer any questions.
Yeah, so thank you very much, Ben. Do we have a quick question for Ben? Okay. So I think these two talks really showcase very nicely what can be done with short gradients, and also when we have this standardized way of executing short gradients. I really think that especially the new mass specs, they really open up the potential of speeding up things. And I think as people adapt their workflows to kind of really get the maximum out of the short gradients, it's really going to make a big difference. So please join me in thanking our two speakers again, and thank you very much for your attention.